Okay. Octacoral taxonomy, and I will speak on some families, some genera. I mean, demonstrating what is the classical taxonomic approach that is being used by taxonomists nowadays in order to give soft corals names. You know how many messages in the old days, letters, faxes I have received. Hudi, please help us. Give a name to the beasts. Very difficult. Very challenging, very demanding. Okay. Well, this is Israel, and we have the tip of the Red Sea. 11 kilometers of coast, 2 kilometers of reefs. But our neighboring sea, the Red Sea, is the cradle of octacoral research, and certainly the cradle of octacoral taxonomy. The reefs are with fantastic reputation, you know, millions of tourists coming to visit Eilat, Egypt, the Saudi coast, etc., etc. But the beginning was with an iconic name, Per Foscal, who on the 18th century went to Egypt to study the pyramids and the ancient scripts and the fauna and the flora of the desert. And somehow everything is from Wikipedia, right? And at a certain stage, he arrived to Jeddah, what is called Jeddah today, and to Yemen, and he collected animals, collected marine elements. He collected the first soft corals ever collected, as far as we know, in the Red Sea, and identified them. Sadly, you can see that Foscal passed away at the age of 31. He was a student of whom, you know? Linnaeus. These Swedish people are crazy, going to the desert. Yes, but unfortunately, he got malaria and he passed away, I think, in Yemen. His collection moved back Sweden, Copenhagen, to Scandinavia. To the best of my knowledge, none of his original colonies, octacoral colonies, still exist. And types were established. And these are the first octacorals ever described from coral reefs. So this was the beginning, long time ago. And since then, because of the close vicinity of Europe to the Red Sea, lots of expeditions and collections were made since then. All these big names, Ehrenberg, Klunzinger, Lamarck, I will mention his name later on, and others, collected Red Sea stuff. It was the closest reefs to Europe. At that time, no flights. Even the Suez Canal was not open. At the beginning, they had to arrive to Port Said on the Mediterranean, riding a camel, no air condition, no ice cubes, no Coca-Cola. You can imagine? Look, Foscal paid his life under those extreme conditions. Famous polar expedition of the early 20s. Well, This you have heard from Eric and from others during the last days. Octocoral, second most important benthic group on reefs, replacing stony corals after catastrophes, invasive spreaders, resilient to global change, lead to shift in coral reef community structure. Well, maybe on the top of everything, bioresource from natural products and biomaterials, well known all over the world. And as an umbrella to our workshop here, spiritual, if you want, mental umbrella, is the recent paper of my colleague. Yes, my colleagues, Katie McFadden and Andrea Quattrini, and my late dear friend, Lane Van Overhen, who passed away about 2 years ago. And we know we are speaking about a new class established in this paper to the phylum Cnidaria, Octocoralia, and the soft corals. Even for me, it's hard to pronounce this. Mele Alcyonacea. Yes, following the Greek word malakos, which is soft. I just copied from Katie's paper. It's not my Greek knowledge. Okay, and we have there like 46 different families. When we speak about soft corals or fleshy octocorals, we speak about organisms which are highly abundant on shallow reefs down to 100 meters plus minus, highly common throughout the entire, warm waters, the Indo-Pacific, not in the Caribbean so, but as you can see, and it has already been mentioned here, the coral triangle is highly rich with those, the Great Barrier Reef, the east coast of Africa, and the Red Sea itself, etc., etc. But you know that octocorals are common elsewhere, not necessarily only in the tropical belt, so to speak. Now, what are the morphological characters used for octocoral taxonomy? I think in a way, Eric mentioned it. But please be aware that I trust morphology. Yes, I know, I am a bit behind. I am a lot behind the molecular and the genetic stuff. Yes, I am. But I think that morphology is something that should be appreciated. Because at the end of the day, once you get a sequence, you want to give it a name. And the name doesn't come with a printer which is attached to the sequencer. You need a bloody taxonomist who is looking for the growth form and ask himself whether the colony is encrusting, lobate, mushroom, branch, etc. A colony has a base and a polypary. The polypary is the part of the colony which bear the polyps. And it has sclerites. They are internal, they are microscopic, their shapes is highly variable. We should be aware about their size range and microstructure. It's not only what you see under the light microscope. Sometimes you have to use power microscopy. The sclerite position in the colony is critical for identification and recognizing the taxa. It's either the base of the colony, the top of the colony, the outer surface of the colony, what some people call skin, but there is no skin here. It's just the outer surface. The inner part, polyps and tentacles. And the last two, polyps and tentacles, normally are small, tiny sclerites that in most old descriptions were not depicted because they are easily washed away. And who cares about these small things? They're like dust. Polyps can be monomorphic, dimorphic, And yes, we found that the symbiotic status of the colony, I mean, under normal conditions, of course, not because of bleaching, is critical, which means the colonies can be either zooxanthellate or azooxanthellate. By the way, you know that the term zooxanthella, it's not valid anymore, but we still maintain it. And we have this trilingual illustrated glossary, which was published like about 40 years ago, maybe more, 82 if I remember correctly. It has to be updated. Yes, we had a workshop in Eilat a month ago, and I told, well, to the participants, take the initiative. It must be updated. This is a reference which is excellent, but we got so many terms that should be revised or added to this glossary, so it will be useful as it was in the old days. Now, you know that classical taxonomy is based on morphological characters. At the end of the road, it leads to assignment of a morphospecies. This is the term, morphospecies. Nothing is wrong about it. Yes, but nowadays, we get initially lots of genetic signatures, and the material is assigned to molecular operational taxonomic units, MOTUs, but not yet their morphology. Therefore, what we adopt nowadays is reverse taxonomy. In other words, Katie McFadden, professor, she gets sequences, and then she sent me a table, an Excel sheet, and she says, okay, Udi, now it's your challenge. MOTU1, all of them are genetically identical. Check their morphology. Yes, she's very, yes, I accept it. I love to identify. Check whether they're identical. I'm looking, checking the sclerites, and say, Katie, hey, number 10 is different. Then we start a fight. Then we start a discussion. Alternatively, all of them the same. Now, well, this is what is written here. Look for congruence between genetic and morphological findings. Now you have to give it a name. By the time that I say that all the MOTUs are identical, I have no name. I have to go to the literature. Right, Daniel? Yes, this is the way. So I go to the literature, and if I am lucky, I get a nice match. And I give a name. Mission accomplished, huh? If not, the problem starts. Right, Megan? And here, no sequence will help. No next generation sequence will help me. There is one solution. I have to go to the type. You know what are the types? The types were designated by taxonomists, normally in the old days, where no sequence was attached to the description, and no scanning electron microscopy was done, and maybe even not the text is, the text doesn't provide the required information. So the taxonomist is getting into a problem and he can't provide a name. Well, in some other cases, he can. And what happens if the type does not exist? He has no reference. So this is the beginning of a symphony which taxonomists are dealing with, including those who are doing octachoral taxonomy. So having the sequences is very helpful for comparative purposes, but this is not the end of the road. Why it's so complex? And every text says that octachoral taxonomy is difficult. is even frustrating because we look for sclerites. And look, these are spindles. They have a term from a single colony, actually from a tiny piece removed from a colony. And you see that there are no two sclerites which are exactly the same. No two sclerites which are identicals. None like two drops of water. What can I do? I have to be aware about the variation within the colony. Not only variation within a colony, but also a possible variation between what I have in my hand and the type which is coming the closest to the colony I have in my hand. This is the difficult, but this is also the fun. There are some exciting sclerites that nowadays can be presented by scanning electron microscopy. In the past, we had hand drawings or camera lucida. You know what is camera lucida? How to explain it? You have a microscope, then you have a kind of a side arm coming from the microscope attached to the optic system. Once you look with your two eyes on your sclera prep, the image, how to say, is being reflected through this set of prisms into a paper, A4, right, white paper, and you look inside and you see the image and you make a drawing. This is the way all those past drawings were made. If my promotion should be based on the quality of my drawings, I won't be here today. I would have another profession. But with scanning electron microscopy, you get these fantastic images of this, what is termed nowadays figure-8 sclerites. I don't agree so much about the terminology, but it doesn't matter. These are from the polyp, from the tentacles. The size range is microns, dozens of microns to millimeters, and they're extremely diagnostic. Now we will make a kind of a survey, a tour. So some of the more common, more abundant soft corals that can be found in the tropics. You're aware about the organ pipe coral, Tubipora, Tubipora musica. And here you see the name of Linnaeus here. Right? Linnaeus didn't collect the material. Linnaeus described it from the Red Sea based on a colony collected earlier by his student that meanwhile passed away. Okay, this is of Linnaeus. What is so unique about this soft coral that it's hermatypic, reef builder? Well, there are some sites that I have been visited, like in the Red Sea and elsewhere, where there are reefs like the size of this classroom and maybe larger, which are made mainly of Tubipora. The sclerites of the polyps are like those figure-8s that were demonstrated earlier, but at the base of the polyps, there are spindles and some branched forms that normally they fuse together and gradually form these tubes. By the way, if you go to worms, you will find there are 7 species, if I remember correctly, of Tubipora. We don't know how to differentiate between them, just names. I know that now there is a student at Ames in Australia who is trying to do it. The most fascinating family, new family established by Katie is the Sarcophytidae. In the past, it belongs, the genera that now are under this family were of the Alcyonidae. You know, the Alcyonium digitatum is a cold water species found in the, in the fjords, the British water, British environment. It's not tropical. So we had the feeling that they cannot accommodate together in the same family with the tropical one. Here you see a kind of a field of sarcophyton, picture taken in Taiwan. It's the most dominant family on Indo-Pacific reefs. Every reef that you visit, almost every reef in the shallow water has lots of sarcophytidae. Sarcophyton itself, type locality is not the Red Sea, for a change. Well, it's a mushroom-like colony as presented here. You have a disc, this is the polypary, this is the stalk, can be large, like 10 centimeters, can be even much larger, diameter of 80 centimeters, even 1 meter. It's dimorphic, which means it has two types of polyps, as Eric mentioned earlier. The colony is fleshy and it requires circulation of water inside its internal cavities. So you have, next to the feeding polyps, you have the siphonoglyphs, which pump in and out water into the colony. All right, what about their sclerites? The sclerites actually are of three types. There are clubs on the surface. A club is typical, wide head, and a pointed lower tip. This is a club. A range of clubs on the surface of the disc, on the surface of the stalk. Inside the disc, the fleshy disc, there are mainly very smooth spindles. When they are very smooth, we might call them, sorry, it should be needles. They are needles, okay? In the interior of the stalk, you get warty spindles. And there are lots of them. I mean, structurally, all of them are embedded in the mesenchyme, providing mechanical strength to the structure, okay, to the colony. Here you see a sarcophyton seascape. The colony is undergoing asexual division, actually fission, and you have at a given site lots of colonies. All of them are zooxanthellate, of course. And you see that these guys are also aggressive towards stony corals. Here you see an acropora, a plate coral on the left, and you see the guy next to it is actually avoiding the growth of the colony, and on the contrary, somehow, damaging the edge of the acropora colony is also quite common on the reef. A kind of a sister genus to sarcophyton, it's lobophyton. Lobophyton is also dimorphic, but instead of having colonies which are mushroom-like, they have, for many species, they have flat lobes. This is why the name is lobophyton. Mostly they are arranged in a radial form, not always, and the polyps are scattered around it. The coloration is due to presence of symbiotic algae in the tissue. So you might ask, what is the difference between lobophyton and sarcophyton, except for the colony morphology? Yes, the sclerites. Typically, lobophyton has in the interior of its base, it has capstans. This morphology is capstan morphology. In the interior of the lobes, there are spindles. So what you get from here, the message that identification is based on a combination of sclerites, not on a typical type of sclerites. Okay? If you want me to make a break, let me know. Okay. You want me to finish as fast? All right, I got the point. Okay. Okay, some of the lobos have no lobes. Yes, that's life. And the name tells Lobophytum depressum. You see the flattened morphology. And here is the name of the hero, Madame Tixier Duriveau, a French expert and taxonomist who worked here, right? I think she passed away in the 70s sometime. Yeah, late 70s. She described lots of new species from the French territories. Her contribution is amazing. You go to the zootech, you count the labels where it's written, Madame Tixier can become a millionaire if each one would be €1. Amazing contribution from New Caledonia, from Madagascar, from Fiji, Atlantic, Red Sea, Gulf of Suez, South Africa, everywhere, everywhere, everywhere. The only problem is that the text is in French. Nowadays, I can use Google, but you can Google Translator, but you can imagine what was in the past. Yeah, but let's go back to science. Well, this enigmatic lobophyton with no lobes. How you can survive with that? We can't. This is a problematic group. There are other lobophytons which reminding, a sarco and not a lobo. Genetically, we have no resolution at the moment. Luckily, morphologically, the sclerites are quite unique for that one compared to the other species, now accommodated, now assigned to the genus Lobophyton. So genetics still waiting for some advancement. Morphologically, it's all right. Different. Now, this is the champion, Sclerophyton, with more than 180 species. Used to be named Sinularia until Katie published her paper. If you would have known how many times I have said in my career Sinularia, now I have to make a switch in my rotten mind. Yes, Mr. Pratt identified at the beginning of the 19th century a colony, and he named it Sclerophyton. And now it has been adapted and replacing Sinularia. And you may ask what happened to the poor Sinularia. The poor Sinularia identified by May in 1998, Sinularia brassica is the only Sinularia remaining out of 180. We were aware about that by morphology. I won't get you into the same images, but if you will see the clubs of Sinularia brassica, you would say, you taxonomists are crazy. It's not Sinularia. And there were rumors in our club of experts that these Sinularia should be separated from the rest. But because it was established in the year 1998, it got the priority to remain Sinularia, and the rest moved to Mr. Pratt, Sclerophyton. So everything has been changed easily on the, on worms, just by click on the computer. And now all collections with hundreds bottles of Sinularia should be relabeled and maybe renamed. But the good news is that genetics discovered that there are more sister Sinularia species. So Sinularia brassica of May won't remain alone in the club. There are more to come. I should contribute my knowledge to that. Some plates are already made. Okay? So you got the point. This is the most diverse and abundant genus on Indo-Pacific reefs, zooxanthellate. Colonies are stocked or encrusting lots of lobes with polyps. The sclerites, clubs of different shapes. The beauty of creation. Just think, 185 species with different clubs. Oh my gosh. Yes, they exist, different clubs. And spindles. In the interior part, yes. Genetics is quite straightforward. Morphology, yes, giving you good direction. A new family established by Katie for a wonderful coral which I love. Cladiella, which was previously assigned to the Alcyonidae, now is within the Cladilidae, a new family. Lobate colonies looking underwater for the non-expert, like Sinularia, the same morphology. I wanted to say, when you were dealing here on gorgonians, there is a major difference between gorgonians, especially the deep ones, and the shallow water octocorals. The morphology of the deep water, upright growth, fan-like structures, getting rid from the boundary layer of the bottom, enjoying the water, and being able to filter the goodies from the water. Shallow water soft corals are mostly flattened, getting their surface area by compound structure, lobes folded, etc., harboring lots of symbiotic algae exposed to the sunlight. Two different strategies. Nowadays, well, it's not taxonomy, but nowadays we know that what limits the depth distribution of these zooxanthellate is lack of adaptability to change their mode of feeding from autotrophy to heterotrophy. They can't do it. And this is the limit why we don't find zooxanthellate octocorals below 65 to 70 meters. I found it first in Eilat, and then we gathered the information from all our colleagues. Yes, they told me, Hudi, no Sinularia, no Sarcophyton below, Sclerophyton below 60 meters. Not on the Great Barrier Reef, not in Madagascar, even in the most, clear and nice water. All right, Cladiella. This is Cladiella, zooxanthellate encrusting lobate polypary, mostly shallow water, but look, the sclerites, totally different. No clubs, no spindles. Instead, we get in the polyp all kind of platelets, including figure 8, sort of, and the interior has dumbbells. You know what is dumbbells, right? Good for your exercise. Look, fantastic. I love those. Now I'm revising the Red Sea ones, all described by Madame Texier, 24 species. They have different dumbbells, different polyp sclerites, amazing architecture. The size is like 0.1 millimeter, more or less, a bit more. Okay? Cladiella on the reeflet, yes. One of the advantages when you dive, you want to give, a name while you are underwater, at least to the genus level. So you touch the colony, you feel its texture. What is so nice about Cladiella, when the polyps retract, the colonies become almost white, grayish. This won't happen to any Sinularia. They are looking alike, but not after polyp retraction. When I see these, I know Cladiella. I mentioned to you, I love them. Another family, Lemnalidae. And here, Katie accommodated different growth forms, different scleroids. But we have to follow genetics. Normally, We are the classical taxonomists, say, don't argue with DNA. Forensic evidence. Rightisma was established by our friend Phil Alderslade about 20 years ago. It's an encrusting colony. Just to remind you, this is the typical surface border in the Red Sea. Flat colonies, membrane-like colonies, encrusting, covering the reef structure or blades of seagrass, etc., etc. Well, this is the original, this is the plate provided by Phil in his book that Eric mentioned, but now we are revising the genus. And we found that in the tentacles, there are all kinds of rods, figure 8, like, I have to change it. It's not figure 8, because we know that 8 has 2 windows, right? Here I can't see any window. But these are actually kind of, okay, in the polyps, and there are spindles quite large. Some of them may be 1 or even 2 millimeter in length. Rightisma fulvum of Forscal was described from the entire Indo-Pacific, including by me. A kind of Indo-Pacific distribution. Fault. Due to DNA, due to the approach that provided MOTUs to the morphology expert. Now we are going to publish 8 species of Rightisma with no overlap in their distribution. And this pattern of biogeographic isolation between congeneric is the pattern that gradually we establish for each soft coral genus. Why one by one? Well, I mentioned to you that the Red Sea was the beginning, the start, the new species were described there. So the mistake was obvious. There was some resemblance, so the ID given followed the original type from the Red Sea, but it was a mistake. And now we are correcting it genus by genus. There are very few pan-Indo-Pacific species so far. So you have for each region its own good species, and I'm happy about it. Okay, within that family of Rightisma, we have another genus. It's not so common, but it exists, Paralemnalia. Type locality is the Red Sea, following the polar expedition, identified by Kickenthal. Well, the lobes resembling asparagus, with some imagination, huh? The fact you have a membrane-like Rightisma colonies and Paralemnalia, the lobate one, a different genus under the same family, it's something that we have to accept. Genetics tell it. Despite the morphological differences between the two. Unfortunately, I couldn't find my scanning electron micrographs of Paralemnalia, but it has these funny, delicate sclerites. Let's skip it. Yes, another big family, very diverse, it's the Neptaidae. And again, what happened here, Katie took it and sliced it. Yes, let's be happy. Now there is order based on genetics, not on morphology that can easily mislead some of us. And here in the Neptide, we have fantastic colored colonies. You know, Dendroneptia. Is it the first one I'm going to show? Yes, Dendroneptia. Also, type locality of the genus is the Red Sea, following Kickenthals, which means the polar expedition. Azozantellate, spiky colonies, depth distribution. When I did my mesophotic, surveys, I expected to see lots of them. No. They're mainly down to 60, 70, 80 meters, then they almost disappear, unless there are some places not explored yet. The spikiness of the colony is due to the polyp sclerites. Each polyp, I hope that you can see it from the back, has supporting bundle of sclerites sitting like that. Large ones. According to worms, there are 200 and plus, 200, I think, 40 species. Nobody hears it. It's between us. Nonsense. The problem is the types. Most of the types got lost. Most of the types were somehow stored in Calcutta Museum, some in Seto Marine Station in Japan. I saw some bottles. No type is there. Maybe the label.
Empty bottles.
Sediment of sclerites at the bottom. No specimens. Probably preserved in formalin, and then the colony got totally. Well, there are more types, like here. It's a project that should be restarted from the beginning, and I believe that the number of species is much lower. Here you see the spindles. It's a spiky colony. By the way, we published a paper in Science, 1995, providing the evidence, experimental evidence, that Dendroneptia is feeding on phytoplankton, being a grazer. The first evidence of an octocoral using this type of food being filtered from the seawater. My colleague Katerina Fabricius, the co-author of Phil Alderslade in his book, it was part of her PhD. She did it in Eilat. She traveled all the way from the Great Barrier Reef to Eilat to do experimental work. Very easy. Beautiful conditions not almost, all year round. No hurricanes, no typhoons, no nothing. Just jump into the water, collect your animals. All right. Dendroneptia is very, very colorful. Its distribution is a bit beyond the tropics. This image I took from Jeju Island. Jeju Island is South Korea, just at the northern tip of the Karyushu warm current, going from the tropics, from the Indonesian archipelago, all the way to Okinawa, to the Ryukyu archipelago, hitting the southern tip of Jeju Island, which is South Korea, about 32 degrees, if I remember correctly, latitude. Now I will tell you a secret. Dendroneptia, which is an iconic genus for coral reefs, just few months ago has been discovered as an invader to the Eastern Mediterranean. Now, two competitive papers were submitted, one by my group, another one by another group. Hopefully that ours will be accepted first, okay? It won't change much for my career, but I have a young co-author, so I want him to get the priority. For me, it's okay. But this is when we speak about octocorals as invasives. Not only an invasive, it's an excellent spreader. Once it's there, it's not a single colony. Hundreds of colonies. And we have records, photographic records of the same spot, taken less than a year before. None was there. Now it's covered with Dendronephtha's colonies. It's a fast grower. It has a watermelon strategy, not much tissue, but lots of what? Water inside. So it's growing fast. It reproduces asexually, sexually in the Red Sea. Massively, successfully colonize artificial structures. And the rest will appear in the paper, okay? A sister genus in the family Nepteidae is Litophyton. Again, 4 skulls collected years ago, identified by Linnaeus after the passing away of the collector. You see, 1775. It's a zooxanthellate branched flabby colony. It has unique sclerites, which are called caterpillars. The spikes are asymmetrically arranged along the sclerites. All right. Here you see Litophyton, large colonies. When some people want to insult me, they call it broccoli coral. No, it's not broccoli. It's good Litophyton. Now you see the 2 genera going one next to the other, same shallow water. The azooxanthellate Dendronephthia and the zooxanthellate Litophyton. Both of them enjoy water currents. When the water are below a certain speed, then the colonies are actually shrinking, having no advantage by being exposed. They are not fully inflated. When the water current is too strong, they don't want to fight with the current. It's the same what we are doing as divers. We don't try to fight with the current, no way. So they collapse, they shrink. The advantage for Dendronephthia is to have a habitat with a water current, which is, let's say, like 25 centimeters per second, and then they can filter. The filtering device can withstand the water strength, the water current, okay? Now, this will be the aperitif. Cnidaires, lovely family. Type locality, Red Sea. The kosher ones, you know what is kosher. The kosher Red Sea. What is so nice that they have pinners, but normally in high numbers. This is a drawing from the dorsal side of the tentacle. You see that you can count the pinners, right? If you look at the tentacles from the oral side, this is the dorsal, this is the oral. You see that there are several rows, in this case, 3 to 4 rows, 3 rows of pinners along the edges. We used to count them because they were considered to be taxonomically important, having at last a quantitative character for octocoral identification. What a pleasure. I have to discourage you. Nonsense. It has no taxonomic value whatsoever. I wish I will have long years to live, long years to live, as much I have counted Cnidaires pinners. Useless. We lost. Xenia, the type of the family. In Latin it's Xenia. In the US, they call it Xenia. First time I heard it, I got almost heart attack. What is Xenia? But that's the way. Established by Lamarck. Merci beaucoup. Yes, following Napoleon expedition to the Red Sea. Appreciate Napoleon. The sclerites under the light microscope are looking like simple platelets. Normally, the diameter is 20 to 30 microns in diameter. Tiny ones, huh? Just remember that my Ratisma has 1 millimeter spindles. So this one is another dimension. Heteroxenia. Also, type locality Red Sea. Those antellate dimorphic. Same sclerites. Under the light microscope, you don't see any difference whatsoever. It's dimorphic, so you say, okay, it's heteroxenia. Antilia, another genus of Lamarck from the Red Sea, assigned to the same family Cnidaires, has different sclerites. Good for us. There are rods, and under the SEM, they are looking like that. We call it cat's tongue microstructure. Do you have a cat at home?
Yes.
The same, but the size is different. This is like 40 micron, if I remember correctly. There is a scale here, 60. All right, can survive. Okay. Well, if you go to the literature, before 2000, I think, and 10 or 11, you may count 10 different genera of Cnidaires listed here. Don't try to read those names, all having the same sclerites. Not species, genera. It means that the statement that sclerites morphology is genus specific is false. Can't bear it. We can't bear it. Can't be. So we started to look at the microstructure. And then what discovered is the following, that the genus Xenia has sclerites whose surface looks dendritic. And you see it here under the SEM. It's not what you see under the light microscope. Nice, huh? High magnification. You don't see it unless you are 10,000 and above. But we used what we call ECM, environmental scanning electron microscopy, where you can get higher magnifications. All right, so Xenia is clear. My colleague Phil Alderslade asked me to scan Ovabunda sclerite. And then we found a different microstructure, which is familiar to you from my notes about ocean acidification, right? The spheroid of the same size, more or less, constructed from a mass of minute subunits, not dendritic. So, two genera, two different morphologies. We are happy, but we have to make a progress. So, the Ovabunda is M&M microstructure. All my students are aware about that. Now you join the club. Okay. Opa. I found another Xenid identified from the Great Barrier Reef, Australia, specimen deposited at the Natural History Museum, London. Cromwell Road. Collected during the Great Barrier Reef expedition. I did SEM. What is the microstructure here? Dendritic? No. M&M? What is that?
Rice. Rice.
Look, 300,000. Sorry, 30,000. 160,000 times. Rice.
Basmati.
Good. Basmati rice. You think that life is paradise? From the height of my age, I tell you, no. I had a name prepared for that, Orizabunda. Genetic says no. It's the same like another genus, which I will show in a minute. Okay? I told you, life is not paradise. Take it for granted. So the question mark still exists. Trust me, I will make it a genus. All right. Another scanning electron micrograph of a spheroid taken from another zenith deposited at the British Natural History Museum, Cromwell Road, collected during the Sir Moritz Young Great Barrier Reef expedition in the 20s. Look how nicely the microstructure remained. The same size, the microstructure is different, huh? Agree with me? Let's move ahead. Okay, 30,000, 100,000. Nobody ever before used such high magnification for these stupid sclerites. When I give this presentation in the US, it's potato chips type. When I give a presentation here, French fries type. Agree with me? You have no other choice. A good genus came out of it, Cematobunda, chips of cement. All right, so microstructure is significantly important and matches the genetics, which is more important than anything else. So the conclusion about Cnidaires so far that we have polymorphic sclerites, compound microstructure, we need high resolution SEM at the nanometric scale in order to look, to find, to diagnose morphological, taxonomic characters and species denomination. And there are novel sclerites which are considered to be diagnostic. The challenge is to sequence them and to find congruence between the sequences and the morphologies that you detect. And this is the grand finale. It's time to finish, huh? Cespitularia. A nice zenids. You see a nice picture underwater. Colonies are like 15 or so centimeter in diameter, maybe more. Branched, you see, branched. Very common in shallow water throughout the entire Indo-Pacific, from the east coast of Africa all the way wherever you go. And I saw them. I won't go into the problem of assigning, lack of ability to assign any colony to this genus. This is the type is of quarry and guy mouth. Does not exist. Considered lost. Checked it very carefully. Spend a day for that. Couldn't find. The description doesn't make sense that it's a zenith. So we have no idea what material was described by Quarry and guy mouth. It's not a zenith. So there is material under the name Cespitularia, identified by May in 1898, deposited in Hamburg Museum. I saw it. I got a permit to remove piece. He collected it from east coast of Africa, which means Kenya and Tanzania, from where I collected lots of that stuff, and I immediately named it Cespitularia, by mistake, of course. Following May, without knowing that May gave the same name to the animal. I made a comparison between maize colonies and mine, looking the same. Sclerites looking the same. Look on their shape. We call it bizarre. No two identical sclerites. Aggregates, different shapes. On the top of it, May says in his description, no sclerites inside. I had the same colony May had. He never used scanning electron microscope because there was none at that time. So we established a new genus, Conglomeratasclera, and you understand why. It's a conglomerate of sclerites of bizarre shape. No, maybe those are quite identical. And the pattern repeats itself in all colonies that we examine. So this is a kind of a lesson that you have to dig for type material. And this is what I'm doing during the last years, re-examining them. Now, the project, hopefully it will be successful, is to sequence them. Using UCE, this is the challenge of my collaborator, Andrea Cantrini, from the Smithsonian, because then we will have a morphological description, correct one, updated one, next to sequence of an old type. If this will be accomplished, I can assure that the following generation of taxonomists will have quite an easy life. I admired the French heritage of diving. When I was a master's student, I met Jean-Jacques Cousteau in Eilat, in his mission with Calypso. I know. And the only diving mask at that time was the Jean-Jacques Cousteau type. And look, we were diving with no BC. I had a depth gauge, couldn't trust it, but I was young and strong. These days are over 52 years ago, 50 years ago. Thank you.